we've already, introduced the concept of wood, and wood is again going to be made up of this secondary xylem. And there's two main types of wood. You've got heartwood, and then you've got sapwood. Heartwood is going to be, more internal towards the middle of the tree. And this is going to be the kind of non-functional, non-living stuff that doesn't have as much access to water and nutrients, so forth and so on. And so these heartwood, this is, going to be the result of aging. So as the tree gets thicker and thicker and thicker, it doesn't have as easy access to all the water and nutrients. So it's like this kind of non-functioning plant tissue that's involved. And then whereas, in addition to the heartwood, you also have the sapwood. The sapwood is kind of the outer region. If you look at, if you cut a tree in half, the heartwood's going to be in the middle, and the sapwood is going to be a lighter color, and it's going to be towards the external parts of the plant. And so this sapwood is also going to be made up of secondary xylem, except it's going to be functional. So they're both made up of secondary xylem, which is what wood is. It's just in the heartwood, they're going to be non-functional because they're in the middle, the core of the tree, whereas the sapwood is going to be those areas, towards the external part of the tree that are going to be functional. And so a lot of times you get these tree rings. And these tree rings will form, again, as these new layers of xylem and phloem are being added as a result of the vascular cambium. And so if you look at the cross-section of a tree, look at this one first. So we have the bark on the outside, and the bark is made up of the periderm, which includes the parenchyma, the cork, and the cork cambium. And the bark, in addition to the periderm, is made up of the secondary phloem. So these outer two layers, the periderm and the secondary phloem, are what's going to make up the bark. Now, the secondary xylem, which is the most internal layer of the tree, this is going to be where all the wood's at. And the wood is arising from this vascular cambium right here. So if you look at the layers that are in a tree, you're going to have the periderm, then you're going to have the secondary phloem, and that's going to make up the bark. And then if you go one more layer into that, this is you're going to find your vascular cambium. And so again, this is what's giving rise to your xylems and your phloems. And then the actual wood part is going to be the most internal part, this purple region, and that wood is going to be made up of secondary xylem. And so you can see maybe this kind of darker region, this is going to be the heartwood. And the sapwood is going to be this outer region. Again, these are going to have the functional cells, and the heartwood's going to have the non-functional cells. Yeah?
What about the rays?
The rays are there to help. So if you look at the tree, right, you've got all this vasculature going up and down this way. The rays help distribute, it's more vascular bundles basically. The rays help distribute stuff laterally, whereas xylem and phloem, most of it is up and down. But the rays are just another way to get those water and those nutrients and those sugars laterally. And so if you look at the cross section, so again you have your bark right here, made up your periderm and your secondary phloem. You've got your sapwood towards the outside, this lighter layer made up of functioning cells. And then you've got all this heartwood, these dark layers that are going to be these non-functioning cells. And so if you look at this under a microscope, so if you just take a region that's overlapping the heartwood and the sapwood and look at it under a microscope, these are the tree rings, right? So there's a ring, there's a ring, there's a ring. So for each ring, this represents a year of growth. So this tree, based on this section, it's at least 1 year old, at least 2 years old, at least 3 years old, 4, and based on how many rings, this is a pretty big tree, so it's going to be pretty old. But these rings are made up of two different layers. You have the late wood, which is going to appear, and these appear seasonally. So you have this late wood, which is kind of this really, really darker kind of red staining area, which is going to come up during the summer. And then you've got this early wood beneath it that is going to arise in the spring. And so these two types, the late wood and the summer wood, as it goes through a whole season, they'll contribute to the width of the plant as well. And so you can tell, so it's this late wood that creates these dark looking rings. So you can see the rings, and the reason why they're dark is because this late wood is kind of a darker color compared to the early wood. So the rings are actually the late wood, and then the space in between the rings, this part right here, is going to be the early wood. So the dark colored rings are going to be the late wood, and the space in between is going to be the early wood. And so this, and especially once y'all start talking about plants in lab, you're going to be looking at tons of slides like this and saying, identify the secondary xylem, identify the bark, identify the paraderm, identify the vascular bundles, so forth and so on. Questions? All right, so that wraps up 21. You want to do some review questions for bonus points? Yes. All right, which type of meristem is found at the tip of the root? You were first.
That would be the long one. Wait a second, give me a second. Hold on. 4 seconds. Just like apical.
You're close.
Lateral thickens and goes from side to side.
Right.
I thought apical was going from long.
Yeah, the apical. Yeah, the apical is at the top and at the bottom of the roots. And so that's going to contribute to primary growth. Okay. Describe the cell types that make up a plant body, Alexandria.
I forget how you pronounce them.
Give it your best shot.
I don't know. Sclerecoma?
Sclerecoma? Yeah. Sclerecoma?
And,
Parenchyma, yeah. So they all end in "-ima". Just either parenchyma, sclerenchyma, or collenchyma. And I have no idea what "-chyma" means.
It's only baby names on Lori.
Okay. Describe the anatomy of a root, Michelle.
The apical meristem, and it produces the root cap and the root tissues.
Mm-hmm.
And then the zones, you've got the cell division, which is your apical meristem and the root cap.
Mm-hmm.
And the elongation, which it elongates by absorbing water.
Mm-hmm.
The maturation, that's where the cells become specialized.
Right, so you've got your zone of maturation, where cells specialize. You've got your zone of elongation, which these cells absorb water, and that helps contribute to the growth of the roots further into the soil. And then you've got the zone of cell division, which is, again, contributing to the growth of the roots into the soil.
Do we need to know the root hairs or?
Yeah, that's a good point. I forgot to mention the root hairs. So on a plant, you'll have a bunch of roots, but then you'll also have all these tiny little root hairs coming off of the roots. And the purpose of those root hairs is just to increase the surface area. Like we said, the more surface area you have, the more nutrients and the more water you're going to be able to absorb. So these little root hairs are just there to add surface area. All right. What?
No. Let's go over that again.
We need to go over that again?
Yes, please.
Okay. The root hairs or the anatomy of a root? Okay, the anatomy of a root. So, root hairs some more. So we've got a root, right? And there's 3 different zones. This is the zone of maturation. This is the zone of elongation. And then this is the zone of cell division. So the zone of cell division, this is where all that mitosis is going on. And you know mitosis is going on because you have this apical meristem right here. And so the apical meristem is, one of those regions of actively dividing cells that are going to contribute to the primary growth. And so this apical meristem is going to be right above what's called the root cap. And again, the root cap is just going to be there to help protect the integrity of the root as it's moving through the soil. So cell division is occurring in the zone of cell division. In the zone of elongation, you've got all these cells, and these cells will absorb water, and as they absorb water, they get longer. And so that's what pushes the root further down into the soil. So the zone of elongation is going to have cells that are getting longer, or elongating, because they're filling up with water. And then as they fill up with water, they get longer and longer, and then that's what pushes the root further into the soil. And then you have the zone of maturation, which is where, new cells, they got to find out what their job is. So they go to the zone of maturation to become specialized in order to figure out what their job's going to do. It's like, oh, you're going to be a part of the xylem, or you're going to be a part of the phloem, or you're going to be, part of the cork, or whatever. Whatever is that cell is eventually going to become that cell or mature in that zone of maturation. Is that better? Okay. All right. What are some differences between monocots and dicots? So I'm going to call on two people, so two of you can answer two of them, and then the other person can answer. All right. You, my lady.
I'll take pollen structure and seed structure.
Okay. So tell me about each one. So what does the seed look like in a monocot or dicot?
It has one cotyledon.
Cotyledon, yeah.
In the eudicote, it has two.
Right. Okay. And what was the other one?
What did I do?
You said
Pollen.
You said the seed and pollen.
The eudicote has 3 pores and the monocot has 1.
All right, good. And your name is?
Chrissy?
Christy, that's right. Okay. All right. Let's do the other two. It's there. Hold on. The tippity top. Deborah. Sorry, Deborah.
Alright, so in the stem of the monocot, all the vascular bundles are spread out
Uh-huh.
all through it, and then the dicot, all the vascular bundles are in a circle on the edge,
Right.
and then in the root, the monocot, they're all spread out, and then the dicot, the vascular structure.
Right, so the monocots are going to have this taproot, and the dicots are going to have this, oh wait, am I getting it backwards? Sorry. Yeah, the monocots are going to have the fibrous roots, and the dicots are going to have the taproots. And then the vascular bundles, like she said, and the monocots are going to be kind of spread out throughout the stem, and in the dicots, they're going to be in this kind of ring structure. All right, last one. Where does wood come from, Stephanie?
It comes from the xylem with which the vascular cambrium will help create.
What type of xylem?
The primary and then the secondary.
Actually, the wood is just the
Oh,
secondary.
it's just the
Just the
secondary?
secondary xylem.
The primary is just the base structure of the tree.
Right, correct.
Primary is the secondary.
Okay, I will see y'all Monday for chapter 22.
Put up the review.